Okay, now we're coming to the basics of plant tissue culture. The modern plant tissue culture have to be performed under aseptic conditions. The lab in which it is performed is always equipped with filters. So the air that is coming from outside that is throwing the oxygen and other things, it's filtered. Why it is filtered? Filtered for because there are a number of microorganisms as their spores that could be present in the air. And the part of the plant which we take for starting our culture, it's known as explant. So the living material, when we cut a plant, let's suppose a stem tissue or a seed, or we can have a part of a leaf, we have to first clean it. So the first step is we wash it with water several times. And after washing it with water, we actually have to use surface sterilization agent. Surface sterilization agents are antibiotics. They could be antibiotics or they could be antibacterial. For example, ethyl alcohol is used. Ethyl alcohol is a strong desiccating agent. It destroys all fungal and bacterial cells. And subsequently, in most of the cases after ethyl alcohol, we use sodium hypochlorite, H2O2, or calcium hypochlorite is required. Once the surface is clear and the explants are transferred onto a semi-solid culture medium, which means you have the culture medium and there is inclusion of agar. So you can have agar slants in test tubes, or you can have agar in Petri plates. There are also cultures in which you can use liquid medium. So you would let the explants float on the cultured medium, which is liquid, when suspension cultures are designed, because in some cases you need to go for liquid cultures that we will discuss later on. Now, the culture medium are very important. It is very important to have a balanced combination of inorganic salts, the macronutrients, the micronutrients, as well as the vitamins and the plant hormones, because this is key to the organogenesis and the differentiation of explants into plants. Remember, when Haberland tried this with null solution, he failed, and the only reason he failed was because his growth medium was not optimized. There was no vitamins, there was no hormones, there was no organic substitutions. So that's why a culture medium is the most important one. What is an explant? As I discussed earlier, it's a part of the plant taken out and grown in a test tube or in a Petri plate under sterile conditions in a specific, again, in a special nutrient medium. This is very important for the type of purpose we are after in tissue culture. We use a special nutrient medium. Morosuji and Scoop is one of the famous nutrient mediums which is used even these days in plant biotechnology all around the world. But depending from application to application, organogenesis, differentiation to differentiation, we will use different combination of plant. And one of the most tricky and the most important combinations is the amount of the plant growth hormones. Should we have more auxins or more cytokinins or more gibberellins? That we will discuss in the next lectures that are coming soon in the next week, inshallah. So you could see it's again an image, a carrot, and there's a piece of it has been cut. And of course, this step involves a lot of washing and surface sterilization. Then the explant is a nutrient medium. Eventually, it will convert into a callus culture. These cells will start dividing and redividing, and then there will be a loosely mass of cells lying around. We call it callus culture. The singular is callus, the plural is calli. And then using the balanced combination of hormones, plant hormones, this callus culture will convert to an embryoid, and from the embryoid it will convert to an embryo, and then the embryo we will have a small plantlet growing in vitro in our test tubes or in our flasks or in our Petri plates. And from here we will be very carefully culturing it and let it grow in the test tubes. Once it has achieved maturity of sufficient level, these plants will be transferred to the field, and then they will be grown in the field just like any other plants. Okay, plant tissue culture involves two major methods. One is in vitro growth, callus culture and suspension cultures, as I discussed. And then the type of explant, it could be a single cell culture. These days, it's very easy to separate single cells. You know, you must have heard about the word FACS, FACS. It is a very well-known cytometry, flow cytometry technique for isolating single cells and characterizing them on single cell level. You can use shoot, you can use a root, you can use a somatic embryo culture, you can have meristem culture, you can have anther culture, you can have haploid production, you could even have protoplast culture. What is a protoplast? When you have a plant cell, there's one of the major differences between a plant cell and an animal cell is there is a cell wall made of cellulose in plants. But if you use the enzyme cellulase and digest the cell wall of the plant, then there is no cell wall in plant, and that part is known as protoplast. So protoplast culture is also one of the methods of tissue cultures. Then we can have somatic hybridization. We can also have embryo culture, ovule culture, ovary culture, and all these cultures are possible because this is the essence of totipotency. Any part of the plant, you take it, you culture it under aseptic conditions, give it the growth medium with balanced amount of nutrients, with vitamins, organic content, and the perfect combination of hormones, and they will regenerate into the whole plant. Environmental conditions, there are three very important environmental conditions for in vitro plant tissue culture: nutrient medium, aseptic condition, and aeration of the tissues. Nutrient medium, as discussed already, it generally consists of inorganic salts, which could be both macro and micronutrients. Macronutrients are very important in making the backbones of the most of the protein structures, for example, and also nucleic acid. Phosphate, phosphorus is one of the major macronutrients. It's needed for DNA, for RNA. The carbon source, of course, there must be an energy source. Vitamins, very important, like nicotinic acid, thiamine, pyridoxine, amino acid for the synthesis of, for protein synthesis. The growth regulators, growth hormones like auxins, cytokinins, gibberellins, and an optimum pH is very important because if you don't provide them the optimum pH, it will be very difficult. One thing is very important to know here: when we have iron, when we say micro and micronutrients, iron is never given to plant tissue cultures directly. It's either rather given in chelation with EDTA, because if you provide iron directly, it will precipitate and it will also affect the pH of the growth medium. Then aseptic conditions are very important because the growth medium, it contains a carbon source, sugar, and it's one of the favorite of bacteria and fungi. So they will really love to grow. So we have to culture this in such a way that there is no inclusion of bacteria and fungi. They can actually they could not only grow on the sugars inside the growth medium, but they can also grow on the plant tissues or the small callus culture or small platelets. So we have to be working aseptic, really important. The last step of important part of this lecture is aeration. All plants, they need oxygen, so we have to give them oxygen to our culture tissues. Is very important. And first of all, when we give them the oxygen has to come through a number of filters so that all the bacterial and the fungal spores and the viruses are filtered. Secondly, we have to keep an automatic stirring and shaking so that the oxygen gets dissolving and all parts of the growth medium, it has the ample amount and sufficient amount, balanced amount of oxygen. This was all for today's lecture, and I will upload it now. You can have any questions in the interactive sessions that we are going to have tomorrow. Until then, take care. Hope you a very good time. Allah Hafiz.